Welcome to our presentation on cloning. My name is Benedict Duda.
And my name is Paulio Bush. We will begin our presentation with an introduction to the topic, and then continue with the three main types of cloning: molecular, organism, and cell. We will then continue with naturally occurring cloning and Dolly the sheep. We will end our presentation with pros and cons, and finally our sources.
Cloning is to produce a genetically identical copy of an organism. The word clone comes from the Greek and means twig, because a twig is able to grow roots and turn into an independent plant. The first successful cloned animals were frogs in 1928. There are three main types of processes used to clone, which we will discuss later. In Europe and other Western countries, there have been some issues concerning the ethics of cloning. However, the consumption of cloned animals has been legal in the United States since 2008.
Molecular cloning. There are four main steps to molecular cloning, and molecular cloning usually deals with DNA. The first step is fragmentation, which is breaking the DNA into single strands. Then comes ligation, which is gluing the DNA back together in a specific or desired sequence. After that comes transfection, which is adding the new DNA into the cells. And the last and final step is screening, also known as selection, which is selecting the cells where the process was most successful.
Now we will talk about different processes used to clone, starting with cell cloning. To clone cells means to populate more cells from a single one. The second process used to clone larger organisms is called somatic cell nuclear transfer, or SCNT for short. There are several steps taken before the copy of the parent can begin to develop in a mother animal's womb. The first step is to take any somatic cell of the organism that will be cloned and to extract its nucleus, where its entire genetic information is held. Next, the nucleus of an egg cell belonging to an animal of the same species is extracted, leaving the shell of the egg without any genetic information of the donor. After this, the nucleus of the somatic cell is inserted into the shell of the egg cell. Because the nucleus was taken from a somatic cell, it has a complete set of chromosomes identical to the animal is to be cloned. Next, the cell is stimulated to grow. After several divisions under lab conditions, the now formed cell cluster is inserted into a female who will carry out the embryo, now forming in her womb, giving birth to an exact copy of the animal that provided the somatic cell. The last form of creating a clone is organism cloning, which is when a cell grows and divides to create an exact copy of itself. This can occur both naturally or under lab conditions. This is basically asexual reproduction. Here's a diagram showing SCNT with the example of frogs. The two nuclei are extracted, then the somatic cell nucleus is inserted into the egg. The egg will then turn into a tadpole and finally a frog.
Now to natural occurring cloning. The most common naturally occurring cloning as we know it is twins. But also in plants and botany there is some naturally occurring cloning with reproduction in strawberries and redwood trees. Now to Dolly the sheep. Dolly the sheep was the first cloned mammal. She is here on the right side of the picture. This was done at the University of Edinburgh on the 5th of July 1996, the date she was born. Dolly died on the 14th of February 2003. The process that was used to create her was the somatic cell nuclear transfer, which Benedict mentioned before in the presentation. Dolly has three mothers. Since Dolly, more animals were cloned, for example, cats, dogs, wolves, and frogs. Dolly died to lung disease and severe arthritis in 2003.
Since cloning can be a very controversial topic, we will now look at its pros and cons. An important point is that under lab conditions, we can manipulate traits by altering or adding or even removing genes. This will allow us to give animals desired abilities. An example of this could be a cow that produces more milk. Also, embryonic stem cells could be grown into full organs for those in need, effectively saving lives. Of course, it would be only limited to cloning organs and not full humans. Like mentioned before, cloning could have a great impact on our food production. Reproduction of consumable animals would be a lot faster. Finally, recently extinct species could be returned to Earth, and endangered species could be saved as well.
Personally, I am against cloning, and there are three main points that strongly speak against it. The first one being that it reduces diversity, which is not beneficial for the survival of any species. The second one is that it puts animal rights at stake, because there wouldn't be as much attention going toward the animals at that point, because they are so easily replicated and reproduced. Finally, it could be abused, because with organ harvesting as well as stem cell harvesting of embryos, the methods that are used are very unethical. These are our sources, and thank you for your attention.